So I mentioned this morning that this lab has to do with protein structure, and we haven't really got the protein structure yet. I know there were some questions in the lab quiz that you may not have been ready to deal with. One in particular about the boiling of an enzyme to see if it would still work. If you haven't done it yet, maybe today will help you answer that question, because we haven't really got into protein. We started talking about amino acids, and you may remember from either previous biology or chemistry classes that proteins are pretty complicated. Unlike DNA, for instance, DNA pretty much always has the same structure. You get that double helix, and it just gets longer and longer and longer and longer. Then you can fold up those helices into like bigger ones and stuff like that. But the structure of DNA is pretty much always the same, with a couple of minor changes. But proteins can take just a mind-numbing variety of shapes. So we're going to learn that the shapes of proteins are very, very much dependent on their environment. And so what we would call the primary structure of a protein is its sequence. That's just the sequence of amino acids. So, glycine, valine, aspartic acid, just all in a row. That's called the primary structure. So we can just read that off of a list. We can just look and see what the sequence is. We can read the DNA sequence of a gene and determine what the primary sequence of the protein is going to be. Next, amino acids close to each other will start to form what's called secondary structures. So these are sort of structures like an alpha helix or a beta sheet or something like that, where amino acids that are really close to each other form a localized structure, like a helix or a sheet. And that is called secondary because it's all sort of localized. So when a protein starts to fold, the secondary structure starts first because they're all amino acids that are really close to each other. And then once you have all these little parts of structure around the protein, then the whole thing folds into a complex three-dimensional shape. And that would be the tertiary structure, when you have the overall shape of a protein. You have a definite inside, you have a definite outside, and the protein now has the shape it needs to do whatever the job is. Be if it's an enzyme, it can start catalyzing those reactions. If it's something like hemoglobin, it can now start carrying and transporting iron around. But none of that's possible until it takes up its final shape. So what we're going to be doing today is looking at the structure of protein in milk, the main protein in milk called casein. And we know there's, protein, milk is a great source of protein and lactose, as we found out this morning. But the structure of casein is really, really interesting. And so I sort of want to go over that with you today, because it's going to be important to how we do the experiment. So we're going to take milk, and we're given some non-fat dried milk powder. And that'll help because if there's a lot of fat in the milk, that will change the structure of the protein casein, and we will see why as we sort of go into what casein looks like. And we're going to learn about something called the isoelectric point. And the isoelectric point is like really, really important for proteins, because most proteins are least soluble at their isoelectric point. I want you to think about that point, because when we start to explain what isoelectric point means, may help you better understand why that's the case, why they're so, why the structure of protein is sort of not very good at the isoelectric point. It's the way that its shape can fall apart at the isoelectric point. So we're going to figure out what the isoelectric point of casein. We're going to measure the pH of milk before and after acid addition. We're going to determine what the pH is at the isoelectric point of casein. And we will see that at the isoelectric point of casein, it precipitates. It will fall out of solution. So we're going to test the milk that's been provided. And then on your own, you will look at another kind of milk. You can use like sheep's milk or cow milk or whatever kind of milk you might have in the house. Try and use low fat if possible. You'll get slightly better results. And then we're going to try and determine what the protein content of our milk is. About 80% of the protein in cow's milk is casein. So you can see that that's like pretty much the main source. And interestingly, casein, let me see if I can draw on this thing. Casein is not terribly soluble, which is kind of surprising when you think about it. You think that if you have all this protein in there, you would want it to be soluble and hang around in the milk. But actually, it's really quite insoluble or poorly soluble. It's really hydrophobic. So what do I mean by it's very hydrophobic?
It's not soluble in water, water-fearing.
Water what?
Water-fearing.
Water-fearing is just terribly frightened of water. Yeah, it means hydrophobic would basically mean that a lot of I think we sort of talked a little bit about amino acids. We'll talk about them more when we get to them. But basically, amino acids have an acid part. They have a carboxylic acid. They have an amino part. And these two join together to make an amide bond. That's what keeps proteins together. But the other part of the molecule, we have this sort of, R part of the molecule, is what's called the functional part of the amino acid. It has side chains on it that do things. And so the side chains are basically what would be exposed either to the interior or to the exterior. And if the side chains are polar, where would you think we would find them? Would we find polar side chains on the exterior or the interior? So polar would mean like it has groups like, it has carboxylic acid group, or it has another amine group, or it has hydroxyl groups, or it has sulfhydryl groups, things like that. So would you find those on the outside of a protein or on the inside of a protein? What do you think? People haven't read the lab. So what's going to be on the outside of the protein? Is the outside going to be exposed to water or not?
Exposed to water.
Hydrophilic.
Yes. Right. So
it's outside.
So if the exterior of the protein is surrounded by water, what kind of side chains should we see on the outside? Would they be polar or non-polar?
Polar.
Polar, exactly. We would want polar groups to be on the outside because they can make hydrogen bonds with the water, make the protein more soluble. So the more interactions that the exterior of the protein can make with water molecules, the better, because that makes it soluble. It's interacting with the water, and it keeps it in solution. Now, there are also non-polar side chains that basically have nothing but long chain alkyl chains, like, long hydrophobic chains that are just carbon and hydrogen. They're just hydrocarbon side chains. And those tend to be buried in the inside. Why? Because they can form London dispersion force interactions with each other and and maintain the stability of the interior. There will be some polar molecules in the interior that interact with each other, like you will see acids and bases sort of interacting in interior sometimes, but generally the interior of the protein is not going to be a lot of water around. There'll be some, but there'll be far more water on the outside. So usually you'll see the hydrophobic amino acids buried in the interior of the protein, and polar side chains being on the outside interacting with water. But casein is really hydrophobic. It has a lot more hydrophobic amino acids than it does polar amino acids. So it's right on the line of being sort of soluble or insoluble. And basically what you find is that milk, the protein in milk, is actually part of a suspension. It's an undissolved protein in a suspension of really, really, really tiny particles that are so small that they float around in the milk and you don't really notice them. But if you've ever left like a glass of milk standing for like a couple days, you may notice that it starts to precipitate, and you will see water on sort of like a cloudy water on top, and sort of like a proteinaceous kind of goo on the bottom. It will eventually separate. And if anyone's ever boiled milk, you'll see that that happens really quickly. If you boil milk, that tends to destabilize proteins, and then they sort of unfold and then crash. Because what happens when you unfold a protein, basically then you expose the exterior of the interior of the protein to the surface, and the interior doesn't make very many interactions with the water, and so the whole thing just sort of crashes out of solution because then it isn't soluble. The interesting thing is that the exact structure of these bubbles, or called micelles, isn't really understood all that well. So you remember we talked about micelles when we talked about making soap. The micelle has an interior of hydrophobic interactions and an exterior of hydrophilic interactions. And that's what's happening here. We have these little bubbles of protein with their interior being hydrophobic interactions, hydrophobic amino acids in the middle, and around the outside has polar molecules. So one kind of casein is called kappa casein. And they're arranged in such a way that you would find in a typical micelle. You have polar or charged amino acids around the outside, along with calcium and phosphate ions. Calcium and phosphate ions are attracted to these charged amino acids and help stabilize the micelle. And that's a good thing because we know that milk is also a good source of calcium too. So it's the calcium that's helping the micelles maintain their structure. And so this is actually what the structure is thought to look like. No, excuse me. And so it's interesting that it's called the hairy layer. I think that's hysterical. So so here we have sort of long amino acid chains that are polar on the end. So these would be like carboxylic acid groups and amino groups and hydroxyl groups on the outside. And then we have calcium phosphate ions embedded in the micelle as well. And then we have these different caseins, 2 different alpha caseins and a beta casein. And all these proteins come together and form this big sort of, as you can see, amorphous kind of lump. So these things aren't really regular. They're sort of just thrown together and try and maintain, oh, excuse me, I have to get some water, and maintain as much stability as they can. So you have the polar regions on the outside interacting with water molecules and calcium phosphate, and then the interior basically is where all your hydrophobic side chains are buried. So you can separate these particles by changing the pH of the milk. And what we're going to be doing is adding acetic acid. And when we add acetic acid, what's going to happen to the pH? Is it going to go up or is it going to go down?
Down.
Down, right? It's going to decrease. So I think that I can't remember, the pH of milk, I think is slightly acidic, lightly. Or it's pretty close to 7, but I mean, we'll do that experiment. But by adding acetic acid, the pH is going to go down. And what we'll find is that the addition of that acid disrupts those intermolecular forces that hold the micelles together, because you've got charges, you've got negative charges and you've got positive charges. The acidic amino acids are going to be negatively charged, and the basic amino acids are going to be positively charged. So you've got these 2 things being charged. And as you change the pH, what you're doing is you're going to be pulling off protons from the basic amino acids and making them neutral. So they'll be going from a positive charge to no charge. And you're going to be adding protons to the acidic and turning them, having them have no charge either. So you're going to be changing the overall charge. I'm sorry. So yeah, the more acidic you get, the less charge you'll have on the acidic ones, and actually the more charge you will have on the basic ones. Sorry, because yeah, when you increase the concentration of H+, more and more of the basic amino acids will get charged with a positive charge. And as you increase the positive charge of protons, you have a negative charge on the acidic ones, and then they'll become neutral. And at some point, at the isoelectric point, basically the overall charge of the protein will be 0. So basically you'll have exactly the same number of negative charges and exactly the same number of positive charges. So the overall charge of the protein will be 0. That doesn't mean there'll be 0 charge. It just means that the positive charges, the negative charges will be identical. And that helps basically destroy these interactions between positive and negative charges that were sort of holding the protein together. You're going to be destroying those interactions by changing the charge of one or both of them. And so that'll break up what's called a salt bridge. A salt bridge is where you have a salt is basically a positive charge and a negative charge coming together. That's a salt, right? We have a positively charged metal and a negatively charged non-metal. Well, in protein structures, we call that a salt bridge. When we have a positively charged amino acid group and a negatively charged amino acid group, they come together and form what we call a salt bridge, because positive and negative like to hang out. Well, if one of those goes away, then that interaction's broken. So you have all these interactions in the protein that sort of keep this thing together. And when we start breaking them, they fall apart. And then once they start falling apart, basically all of the 3-dimensional or tertiary structure of the protein starts to fall apart as well. Then you get the interior sort of being exposed to the water, which it isn't soluble in. And then the whole thing just starts to denature. It no longer has its natural tertiary structure or three-dimensional shape. And then it just crashes out. So it denatures and then it coagulates together. Basically, you have all these proteins, you have all these hydrophobic structures basically looking for somebody to interact with. And they find another hydrophobic area of another protein, and they stick together and they stick together, and you form these big lumps that all stick together and that all crashes out at the bottom. And so you get these insoluble proteins coagulate, precipitate, and fall out. So when are they going to do that is the question. So it gives you an example of what does isoelectric point mean. So at a certain pH, as pH decreases, so here we have a basic, let's say, here we have an acidic. Whoops, let me go back. Yeah. So here we have the pH going up. So we're going from acidic to basic. So under acidic conditions, we have a lot more protons. So what's going to happen is that our acidic side chains are going to be uncharged because they went from having a negative charge to now having no charge because they have a proton added to them. But the basic side chains are going to have another proton added to them and make that nitrogen positive. So under acidic conditions, we're going to have a net positive charge. You're going to have a lot more of these basic side groups being positive and hardly any of the negative ones, or hardly any of the acidic ones being negative. So we'll have a net positive charge. Under basic conditions, the exact opposite takes place. Under basic conditions, we have more hydroxide ion. So the hydroxide ion is going to pull these protons off and make water. Therefore, turning that basic amino acid back into being uncharged. But what it will also do is pull that proton off of the carboxylic acid and make that negative again. So under basic conditions, we're going to have a net negative charge because you're going to have a lot more negative charges than positive ones. And so we'll have more negative charges. And at some point in between acidic conditions and basic conditions, you're going to have exactly the same number of negative charges and positive charges. And so the net charge will be 0. And it's at that point that things start to go awry and that the protein will start to fall apart. So basically proteins don't like it to be too acidic. They don't like it to be too basic, but at the same time, they don't want the net charge of the protein to be 0 because that's not good for them either. So what we're going to find is that we'll look at what the pH of milk is, and we'll see that at that pH, the protein is most stable. The casein proteins are most stable. When we start to decrease the pH by adding acid, we'll go from a net negative charge. So it's net negative at whatever pH whatever the pH of the milk is. At that pH, the casein will have a net negative charge. And as we start adding more and more protons, it'll get to the point where it gets a net 0 charge, and then it'll crash out of solution. So we're going to drop acetic acid, drop by drop and keep stirring until it no longer forms any more precipitate. And that should be the approximate PI. That's what we call the isoelectric point. That should be the approximate PI of casein. And I'll also look up the PI of casein to see if we're close. So any questions about the isoelectric point?
Yeah, I didn't understand it.
Okay. So let me just clear all this stuff. Here we go. So we know that proteins have negatively charged amino acids and positively charged amino acids. So, here we go. So it has negatively charged amino acids and it has positively charged amino acids. So these are the acidic side chains, and these are the basic side chains. So at a neutral pH, some of these are going to be acidic and some of these are going to be basic. As we change the pH, as we make the pH more and more basic, we increase the concentration of hydroxide ions. And what that does is it pulls off these protons and makes water. So basically, we're going to have the acidic ones are going to stay negative, and the basic ones are going to have no charge on them. So at very, very basic pH, the protein will have a net negative charge. Basically, all the acidic ones will be negatively charged, and all the basic ones will have no charge at all. So overall, the protein will have a net negative charge. At super low pH, where we have lots and lots of protons, that is going to protonate the acidic amino acids. So no longer will they have a negative charge, they will have no charge because we'll have added a proton here. The basic proteins will stay positive because basically the high concentration of protons will protonate the basic amino acids too. So the basic proteins, the basic amino acids will be positive, and the acidic amino acids will have no charge. So at very low pH, we have a net positive charge because the acidic ones have no charge, the basic ones are all positively charged. Somewhere in between, we're going to have exactly the same number of negative charges and positive charges. Those will be identical. And at that pH, the net charge of the protein is 0 because it'll have, in this instance, it's got 2 positives and it's got 2 negatives. We add those together and we get 0. Generally, at that point, where we have a net 0 charge, the protein is at its most unstable because once the protein doesn't have positive charges to interact with the water, or it doesn't have negative charges to interact with the water, it has basically a net 0 charge to interact. And so it becomes the least soluble at that point. You break a lot of these interactions between positive and negative by either adding a charge to one or taking a charge away from the other. And so we break a lot of those interactions and the whole thing kind of falls apart. So what we'll find, since we're adding acid to get to the PIs, that tells us, so we're adding protons. So we're going in the opposite direction. So what that tells us is that we must have had a net negative, casein must have a net negative charge. And that helps to make it soluble. And that makes sense because if we have a net negative charge, we have something for these calcium ions to interact with. So we want a net negative charge for these calcium ions to interact with, to help stabilize the protein. By adding more acid, basically what we're doing is we're taking the charge away from those negative charges. We're adding protons to them, and we're decreasing the number of negative charges. And as we decrease the number of negative charges, eventually they'll equal the number of positive charges, and we'll get to the PI. And then at the PI, the protein is going to precipitate. Does that help? A begrudging thumbs up. We can talk about it. We can talk about it more, and we'll talk about this more in class as well. So how are we going to figure out what the PI is? Well, first of all, we're going to make some milk. So we have some dried milk. So we're going to use this dried milk, and I think we're going to make 100 mils of it. And then we're going to take 10 mils of that and treat it with acid. So we're going to need our acetic acid. I don't know why different colors are different. So here's the acetic acid. There we go. And so we'll be adding that drop by drop and then stirring with our glass stirring rod, unless yours is broken like mine is. So we'll test the pH after we've made the milk. We're going to make the milk in the 250 ml beaker, and then check the pH of it using the same paper we did last week. Once we've checked the pH, we start adding the acetic acid. The pH starts going down. We'll start seeing precipitate form, and we just keep putting drops in and stirring until we no longer are forming any more precipitate. Basically, it looks like we have a relatively clear, it'll still look milky, but it'll be relatively clear solution of water and whatever else is in milk. Then we're basically going to we're going to filter this out. So we have our filtration funnel and some filter paper. We'll weigh the filter paper, and then we'll filter the milk protein through, the casein through, rinse it, and then let it dry, sort of like we did before, and then measure the mass. And it's expected we should get about 3% of the total mass should be casein. So if we have, let's say, 10 grams of milk, what should be the mass of our casein?
Three grams.
That would be 30%.
0.3.
There you go. Yeah, it should be about around 0.3 grams or so. So whatever milk you're going to use, look up and see what the expected protein concentration is. It's probably going to be around the same, but you're going to need to know that. So first we make the milk with 10 grams of dry milk and 100 mils of water. Well, not 100 mils of water. You bring up the total volume to 100 mils, and then just stir until that's completely in solution. Weigh a medicine cup. Add about 10 mils of the rehydrated milk back. Record the mass. That'll give you your mass of milk. And then take the pH, start adding the acetic acid till it stops precipitating. Take the pH again, and then we're going to weigh the filter paper and then filter our protein product. So at the end, what we're going to want is our mass, that the cup we started with, to get the mass of the milk. So this is going to be somewhere probably around a little over 10 grams. We want the pH before adding anything, the pH after everything stopped precipitating, and this should be approximately the PI of casein. Mass of filter paper with dried casein. So you're going to probably let that dry overnight, just like we did before. And then figure out what the mass of casein is, and then your percentage. Like I said, it should have approximately 3%. So on the report, there's room for the dry milk sample that we're going to make today, and then you can do, once you've done that, then you can do the your own milk. If you don't drink milk, then I don't know, find some milk or something, or you can just do the dry milk sample again and then compare the results. But I want you doing it twice at least. Okay. So any questions before we get all of our stuff together? You can see this shouldn't take a whole lot of time. Most of our time will be waiting for our protein to dry, and then take the mass in the morning. Any questions?
So I can do it just 2 times with the dry milk, because I don't drink milk, so I don't have milk. I have almond milk, but I don't think it's gonna work.
No, there's not going to be a lot of casein in almond milk. No. Although it would be interesting to see what kind of, I mean, obviously there's protein in it, just not this kind. Yeah, then you can just do it twice then. That's fine. Anybody else have any really cool milk? Like, you got like goat milk or something? All right.
I have
So-
a question.
Oh, yes.
But on the last lab, is that okay? Or can
Oh,
we?
sure. Yeah.
I don't know if I made my soap wrong, but there's like no foam, like at all. There's no foam.
Oh, it didn't. So
Oh.
you filtered it, and then did you actually make any?
Yeah, I mean there was like a little bit, but it wasn't like a full 0.5 grams.
I noticed that. I noticed mine. I didn't get a great deal either. I don't know whether the vegetable oil is different than the kind, because last term it made a bunch. I don't know if the vegetable oil was different. Yeah, I noticed the same thing. I didn't get half a gram either. I got about 0.2 or so.
Yeah, and like even just trying to shake it, like there was barely like a tiny foam layer. But with the oil, it kind of created a little more. And then with the hard water, there was like no foam at all.
There you go. That's a result.
What does that mean though?
Well, it just means that that it was the soap was much weaker than because it was much less, it was more dilute. So if you have like really dilute soap, obviously it's not going to make nearly as many bubbles. So yeah, so you mean we just make do with what we have. Yeah, I noticed the same thing. So there was something was amiss with that oil, I think.
Okay, thank you.
Yeah. Or maybe we should have done the hot. I mean, maybe heating it would have helped move it along a little bit faster. Yeah, I'll look into that. Okay, so why don't we get back together at 3? So about 15 minutes, we'll get all our stuff together and we'll get started, okay? All right, see you in 15. So to make our milk, we're going to make it in the 250 ml beaker. So I'm just going to put, oh, I got ounces again. I want grams. There we go. And then tare. So we want about 10 grams. Notice it just says 10 grams without even a dot, so pretty much anywhere close to 10 grams is going to be good enough. You want to break this up a little bit because mine was kind of chunky. And I used to have to drink this as a kid. No wonder I liked milk at all. There we go. Wow, 10.01. That should be close enough. So, only filtered water. So bring this up to the 100 ml mark with water. So here's the 100 ml mark. And then in the bag, luckily there was another glass stirrer, which was nice because the other one I had was broken, so I can use this. To get this into solution. But of course, as we've learned, it isn't really in solution, it's in suspension. And that's an important difference. With most proteins, they sort of exist by themselves. There are some that form complexes, and we call that quaternary structure. Then we have 3 or 4 or 5 or 6 proteins come together and form a structure. But this is a lot more haphazard. There isn't a particular number of proteins that come together to form a very specific structure. So it's more of a suspension. This goes into solution pretty quick. The check, yeah, just look and see if there's anything left at the bottom. That's pretty fast. What we want to do is get our pH paper. pH paper. And see what our pH is. So we just get a little bit of milk on the end of our stick here and just sort of drag it along. pH paper. Make sure that's enough. There we go. And then wait a minute. And it looks like it's, you can see it's a little less, it's gone decreased in color a little bit. So that looks to be around pH 6, maybe. Yeah, looks to be around pH 6. So yeah, so when I compare it, the closest pH seems to be around 6 or so. And that's to be expected because one of the things in milk that contributes to the pH is lactic acid. That's where we get the name lactic acid from, is from milk. And so with the presence of an acid in here, we figure the pH is going to be slightly less than 7, and it is. So then next, I'm going to want about 10 mils of that. Here I got my 10 mils. And again, you don't need to be super precise, just around 10 mils, because the mass is going to be more important than the volume. So you should have one of these little medicine cups. And we want to get the mass of it. So then I pour it in. And what a surprise, it's around 10 grams. A little more than 10 grams, which we would expect because it's not just water. We've added a whole bunch of other stuff in there, but it's not a lot more. So I got 10.27 grams. So just over 10. So I've got my pH, I've got my mass of milk, got my vinegar here. I'm just going to put the vinegar in a cup just to make it easier to get out. So one thing to remember when you're working in a lab is we would consider this like our stock solution, basically, or actually our working solution. So I'm going to pour a little bit out so it's just easier to use. But what you never, ever, ever, ever do is pour it back into the original container. No matter how much you take out, just dump it, because that leads to your working solution being contaminated with whatever. And you don't want that. And then the other people who work in the lab are going to get really angry at you. And for good reason. So then I get my clean pipette. And I just sort of just start putting drops in here. And I should be stirring as well. So far. I really haven't seen anything start yet. But one thing you'll start to notice is that notice on the sides, you'll start getting proteins being stuck to the sides. That's the protein. Some of the protein is actually starting to precipitate now, just with that little bit of acid in it. And you should also notice that the texture will start to change. Notice it's sort of looking kind of creamy. It means the protein is starting to change its shape. And I'm starting to get protein stuck to the glass stir rod as well, which is probably going to affect your yield. So still in suspension, but definitely starting to look different. Now I'm starting to see little clear areas. Protein's definitely starting to stick together now. Yeah, it's kind of hard to see, but yeah, see how it's sort of starting to clump together, where before it was sort of this smooth texture. Now it's starting to clump together a little bit. Yeah, I'm getting more and more protein stuck to the sides, which is definitely a sign that it's precipitating out of solution. This is a really common thing. I know there's a lot of desserts. There's like an Indian dessert I can think of. I can't remember the name of it, where you precipitate milk solids out with vinegar and then sweeten it. Anyone remember what that's called?
Isn't it kefir? Kefir?
Yeah, something like that. Yeah.
Kefir.
There's a dessert where you actually like form it into balls. It's really yummy. Yeah, kefir is sort of more like a yogurty kind of thing, I think.
Mm.
So it's sort of difficult to determine when this stops. So I'm just going to keep adding a little bit more and see. I mean, I definitely have more of a suspension of larger particles than I did before. But this isn't going to be like when we did copper and it just all crashed out of solution and fell to the bottom. Proteins aren't going to be nearly as dense because they have so much water in them.
I have a question.
Yep.
Since it doesn't give us an exact amount of how much vinegar we should put in, is there such thing as putting too much or too little? Will that affect our results?
Well, yeah, I mean, basically what you're trying to find is what the PI is. So if you add too little, is your pH going to be too low or too high?
It'd be too low.
If you had too little acid,
Oh, sorry, it'd be too high.
that's right. If you had too much vinegar, it would be too low. So, and we're basically, yeah, I mean, this is a home experiment. So we can't be expected to get, but we should be within, I would say, one, like a half of a pH unit, plus or minus. Yeah, but it is difficult to tell whether, like, if I have more precipitate now than I did 3 drops ago. So just for, I mean, just for yield's sake, I would probably err on the side of adding a little bit too much. But yeah, now I'm sort of to get like kind of like a foamy precipitate that looks different than it did just maybe 10 drops ago. But yeah, I'm just going to check on the pH right now. So yeah, so here's the original pH of the milk. And here's what it is now. So it's definitely more acidic. Let's see by how much. Well, it looks like I'm up to about pH, I would say that's pH 4. Probably closest to pH 4, maybe even 3.5 or so. Yeah, so I haven't really noticed any big changes. So probably just like maybe 3 more drops, that'll be the end of it. And then one last check of the pH. And then I'm going to start putting together my filtration. Yeah, I'm getting, it's kind of hard to see, but I am getting larger and larger sort of precipitates there. It looked, the individual pieces seem to be larger than they were. So I might be right where I need to be. Check the pH one more time. Yeah, it hasn't really changed all that much from before. Maybe closer to 3.5 than 4. So I'm just going to let that sit and then start putting my filter paper together. So here's my, so you should have a little funnel in there. And I'm just going to use the 50 ml volumetric to hold it. It just makes a nice Makes a nice stand. So for the paper, just fold it once. Oh, get the mass. Almost forgot. It's going to be kind of hard to get the yield if you don't know the mass of the filter paper, because we're going to measure the mass of the filter paper plus the protein. Much easier than trying to scrape all that stuff off of the filter paper. Keeps resetting itself to ounces for some reason. All right, so 1.27 grams. Pretty much what it was before. There's two different ways you can go with this. You can go sort of like just the regular route, fold it twice. And then that just makes a nice little thing that fits inside. Or if you're feeling daring, you can fold it a few more times. And make a fluted filter. Like this. What I would recommend is wetting this with a little bit of water so that it sticks to the side of the just put a little bit of water in there. See, that way it's when it's wet, it actually stays in place. Oh yeah, definitely. I think we've definitely hit the, you see it's pretty much separated now. Just let it sit for a minute. And then, yeah, rinse out your cup with a little more water because there's going to be a bunch of protein on there. Try and scrape as much of it off as you can. Remember, it's not actually going to go into solution. Maybe a little bit will, but this thing filters really quickly. So and then basically that is it. So then once it's done, we're not going to worry about washing this because we're not particularly concerned about using it for anything else. If there's a little bit of acetic acid in there, really not that big of a deal. So then just take it out and let it dry. overnight. I'm just going to get something to put it on. It's a piece of paper towel. And there you go. Yeah, a nice amount of protein there. So you can actually even flatten the filter paper out and let it so it dries a little easier. So yeah, it's a fairly nice yield there. So start with the mass of the milk, not the volume. Remember to take the mass of the filter paper, and it should be around about 1.3 grams or so. So when you weigh this tomorrow, you should get somewhere between like 1.5 to 1.7 grams or so. And then calculate what the percentage of casein in your original sample. It should be pretty close to 3%. If it's a little bit less or a little bit more, that's fine. But if you get like, 10% or something, that's probably something wrong. So I'm just going to check what the PI of casein is. I think it's definitely acidic, but I can't remember exactly.
Professor,
Yes.
how many drops of vinegar should we use? I'm pretty slow right now. Like, I got it into the medicine cup and then.
Well, apparently I went way over because the PI should be somewhere around 6. And I went to pH 4. So I definitely got all my protein precipitated. So yeah, I would just go like one, instead of going one at a time, I was going like 3 or 4 at a time, which is maybe I was in a rush. So it shouldn't take much more than about 10 or 15 drops, I think.
Okay.
I've never been the most patient of people, so. Yeah, I thought the PI was much less than that. I thought it was around 4.5 or 5, but that was kappa casein. So let's check and see if I just get a different, if I just look for PI of casein. There we go. Yeah, if you look up isoelectric point of casein, it's 4.6. So I had 4. So like I said, I was off by about half a point. So I think that's acceptable. So what I would do is I would check the pH going through. So the pH of the original milk was around 6. So you definitely want to go to 5 and then probably slow down after that. I would check the pH like a couple times as you go. You got lots of paper, so don't. And you could use one end of the paper for one, and then if the other end is dry, you can use the other end as well. So I would check it like 2 or 3 times. But the answer you're looking for is around 4.5. Because if you remember, there wasn't just one protein, right? We had alpha, we had 2 different alpha proteins, we had a beta protein, we had a kappa. So they're all going to have unique PIs. So what we're basically dealing with is what's the lowest one, because at that point, everything will have precipitated. Then you get all this lovely dried milk left over with that awful blue color that I just still gives me the heebie-jeebies. Now that I can afford organic milk, I'm much happier. All right, any questions, folks, about what to do? As you can see, pretty quick experiment. We're really looking for 2 things: the pH and the yield. How you doing, Jessica? You got any questions? You finished?
Yeah, I was
The
just gonna
dry?
say that I think I went too far too, because I also got a pH of 4.
Before? Yeah.
Yeah, but I did.
Well,
did too
again, I mean, this is, again, we're not dealing with, like, the most accurate pHs here. So it could be, you could get something like, 4.3, and you look at this, would you really be able to tell what 4.3 is from this? No. So yeah, so that's why I'm not going to, mark too harshly with a pH 4, as long as you're in the ballpark. If you went to pH 2, then I would think, yeah, you probably, then you just dump the vinegar in. But yeah, anywhere between 4 and 5, I think you're going to be fine.
Okay, so I think I'm safe there.
There you go.
Because it's hard to tell, like, because the colors are pretty close. Like
Oh, there's totally, yeah,
4
yeah.
and 5. So maybe it was 5.
No, you could see
4.5.
it's relatively straightforward to see, like, yeah, 3, 4, and 5, or even, well, no. 3 and 4 appear to be a little different. 4 and 5 look really close.
It's close, yeah.
Yeah. 4 and 5 is like, yeah, if you have color blindness, this is not the lab for you.
I am colorblind for some colors, like like the pH 4, this
Red, yeah, the red.
shade of orange.
Well, yeah, I mean, if you have red-green color blindness, then yeah, this is going to be damn near impossible.
I'm
I
just
think what's frustrating is like the paper starts out as orange
Right. Mm-hmm.
and then it's like, is it working? And you realize like 6 is orange too.
Yeah, if it doesn't do anything, then it's pH 6. Yeah, pretty much. Or I guess pH, yeah, pH 6. It is pH. It's set to basically to pH 6. Is that color? And then when it gets a little green, little tinge of green, then it's 7. And then a little more orange, it's 5. Or a little darker, it's 5. All right, any other questions? Short one today. Dogs get an early walk.
I have a question.
Sure, what's up, Dee Dee?
I have a milk, like cow milk, but it's unpasteurized. Is that gonna work?
Sure.
Oh, okay.
Oh yeah, no problem. No problem. Is it relatively low fat?
I don't think so.
Yeah, give it a try. What the hell? That's
Okay.
the spirit of adventure, right? That's what experimentation is supposed to be. Unpasteurized, man, raw milk. That's hard to find. Where'd you find it?
They have one at Molly Stone.
Oh, okay.
Yeah.
I thought there was wasn't there some legislation about raw? Maybe that was in a different state.
But it tastes like half and half. Actually, it's so much better than pasteurized one.
Oh, it's half and half. Oh my God. So it's like 10%.
But
Well,
I
try
mean,
it. I'm
I fed
really
my,
curious to see what'll happen.
yeah, I'll try. I feed my dog with that. That's why.
You feed your dog half and half?
Not half and half. It's unpasteurized, so it's very thick. I guess there's a lot of protein. It looks like it looks like half and half for me.
Oh, okay. Oh, it
But-
looks like it. Oh, okay. Yeah, well, the pasteurization, I mean, it happens pretty quick. And so the idea of, you wouldn't want to do that for a long time because then all the protein would precipitate. So basically it's heated up like in seconds and then cooled right back down again. So long enough, hopefully, to kill whatever bacteria is in there, but not so long that all the proteins crash out. So it should, I mean, it shouldn't really look that different from regular, but if it's full fat, right? So it'll be like around 3.5% or so. So yeah, I'd be curious because that's going to look different. It'll look radically different. Because if you have a lot of fat in the milk, that's a lot of hydrophobic places for the protein to stick to. And so it may not precipitate quite as much because now you have these droplets of fat in the milk, and the hydrophobic parts of the casein are going to stick to that and stay in solution. So you may find that it'll take a lot more acid to get it to crash out. So yeah, I'm curious.
Okay.
Yeah, I'm curious to see what that would happen. Because I think that's why the recommendation is to use less fat, because if there's nothing but water in the milk, well, then that hydrophobic part of the protein interior has got nobody to stick to except itself, and then it crashes out. But yeah, that's why we do experiments. I'm looking forward to see what your result is. I mean, hell, it may not precipitate at all till you get to like a really low pH.
Are we supposed to keep count how many drops? I lost count.
No one
Okay.
cares how many drops. It's the pH. Check the pH.
Okay,
Yeah. I'm sorry, that was harsh. Someone cares. I care. How about that? I'll always care.
thank you.
All right, folks. So yeah, have fun with this one. It's a pretty short report. And so Thursday, have group meeting. Man, I've only been doing this for 5 years. I keep saying group meeting. Office hours will be at noon tomorrow. And then we'll do the workshop 12 on Thursday. And then I'll come up with something to do next week. I'll be back with you about that. All right, everybody. Have fun.
Thank you.
All right. Bye-bye.